our speaker today. It's Barb Natterson-Horowitz, who really doesn't need much of an introduction for this group. But for those of you who don't know her, she's a professor of medicine and cardiology at UCLA, where she also co-directs the evolutionary medicine program. She also holds faculty appointments at Harvard in both the medical school and in the Department of Human Evolutionary Biology. And she teaches courses at Harvard. She served as president of the International Society for Evolution Medicine and Public Health. And of course, she launched this field known as Zubiquity, gave it its name, and she's had a major impact through her Zubiquity research on understanding and applying concepts from One Health, comparative medicine, and evolutionary medicine more generally. And today she's going to talk about the origins of cardiovascular sex differences, insights from across the tree of life. So welcome, Barb, and the floor is yours.
Awesome. Thanks, Charlie. I'm really excited to be here. I have to say, I don't have a 10-minute. I have about a 20 to 25-minute talk and then a conversation. Amazing. Yeah. So I'll just jump right in. And here we go. There we go. All right. Are you seeing my presentation?
We see the first slide.
Wonderful. Let me hide this and we're good to go. And Charlie, if I should have any technical issues, would you just orally tell me, because I'm not seeing it. My screen is fully slide.
I will. I'll let you know. I'll also let you know if there are any questions that pop into the chat that look like they're simple kind of clarifying questions.
Fantastic.
It's useful to address those as you go. Thank you.
Absolutely. Wonderful. So it's 9:05. My goal is to be done at 9:30, and we can jump into conversation. So I believe we have an opportunity to enter a golden age of evolutionary medicine. And I'm saying that because of the unprecedented amount of biological data that's streaming in. I mean, a diverse, at multiple systems levels, diverse biological data. So much of it is publicly available, and also just the number of genomes that are being sequenced. 70,000, 71,000 NCBI now, and of course the Earth Biogenome Project projects, I think, 1.5 million eukaryotic species sequenced by the end of the decade. So there's this, huge opportunity for us to really move forward. This is, again, it's an opportunity, but for it to be realized, I think we really need to have more methodologies. And we need methodologies that will allow us to generate testable evolutionary hypotheses and validate these hypotheses and really find ways to take evolutionary knowledge and turn it into translational research that can be usable. And then we have to find ways to assess and really test the value of adding this evolutionary knowledge. So these are all methodologies that are needed. I've been really interested in this for quite a few years. It was now just the beginning of the pandemic, so around 2020. I decided to kind of fully focus on this and actually started to work on my PhD and did my defense a few weeks ago. And this is the product of that research, a piece of it anyway, what I'm going to present today. What I have had as my goal is to find ways to really develop phylogenomic and computational methodologies that can generate evolutionary knowledge and integrate it into biomedical research methods. Also to develop methodologies that can really assess, contest the influence of this evolutionary knowledge on biomedical discovery. Now, the second part of the research for the PhD is a machine learning project, which I'm not going to be talking about today. I'm going to really focus on, so this is the broad program, but I'm going to be focusing on some of the methodologies that can generate and test evolutionary hypotheses as a way of integrating that knowledge into research. So I have now, I think, 23 minutes. So let me tell you what I'm going to do. I'm going to offer you a background in terms of why I'm approaching this as I am. Then I'm going to introduce one of the methodologies that allow us to create trade-off hypotheses and test these hypotheses. And then I'm going to show how this methodology can be applied to Takotsubo cardiomyopathy. TTCM is the abbreviation for a form of acute heart failure that is disproportionately impacts women. And I'll go through the results, the implications, and then I'm hoping to, even if we don't have time during this brief lecture, I would be delighted to share the app that I've created. It is intended to be a resource and a tool for evolutionary medicine. I've designed it to have maximal accessibility. It is browser-based, it's interactive, it requires it's a no-code app, so it really is intended to be a tool for investigators with varying levels of computational background. Okay, so a little bit of background. What is evolutionary knowledge? Why is it relevant to human pathology, and how is it identified and applied? You know, this audience is more sophisticated than some of the other audiences I speak to, so please understand that I'm just reviewing things. So, of course, traditional biomedical research is going to ask certain kinds of questions. Evolutionary inquiries are going to ask other questions. Specifically, of course, the question of the typical reductive mechanistic seeking investigator is, what are the mechanisms that are producing the phenotype? Whereas, of course, the evolutionary inquiry is going to ask, what are the selective pressures that have shaped the phenotype? Now, when we're talking about disease, of course, when we're talking about evolutionary medicine, our phenotype is vulnerability to disease. And to the uninitiated, which this group is not, but to the uninitiated, that often creates a moment of cognitive dissonance. Well, how can there be selection for vulnerability to disease? And of course, that's an opportunity for us to go on and explain that not all aspects of phenotype are necessarily adaptive. And of course, this has been well articulated and tested and demonstrated in all kinds of ways. This is just one of the most colorful and memorable. So in essence, when I began approaching this project, I had as a kind of North Star that the trait is vulnerability to disease, and that a consequence of that vulnerability to disease, the trait arises as a consequence of selection for biologically beneficial characteristics. I am going to be using the term trade-off. I'm going to occasionally use the term byproduct. Neither one of those terms, I think, is perfect. I'm happy to discuss that, but I'm going to quickly continue on. All of us are familiar with one disease that is well established as a trade-off, certainly at a population level, of course, sickle cell disease. We are aware that vulnerability to sickle cell disease is a consequence of selection for resistance to malaria, to falciparum malaria, among individuals with sickle cell trait. And this was actually observed in the early 1950s by Allison, when Anthony Allison, when noting that the allele frequency of that the trait frequency was highest in areas where there was significant malaria. And I think everyone is aware of the concept of the heterozygote advantage. In other words, the resistance to malaria, and of course, the homozygosity confers the disease vulnerability. Okay. So this is interesting, and I think every medical school student knows that, maybe even undergraduates. But I began wondering whether we could whether there was an opportunity to look at trade-off hypotheses in a different way. In other words, to really focus on the nature of the adaptive physiology under selection through which the vulnerability to disease emerged. In other words, understanding the selective pressures and adaptive physiology that shaped the emergence of disease vulnerability, whether it could inform our approach to disease itself. And in fact, I believe it can. So one of the things that I did to explore that was I really looked into the mechanisms of resistance to malaria in sickle cell trait individuals. In other words, how is it exactly that individuals with sickle cell trait have this resistance to malaria? And here are just 5 examples: protective effects of hemoglobin, of fetal hemoglobin, inhibition of polymerization of the the hemoglobin, inhibition of cytoadhesion, which sort of inhibits the systemic transfer of the protozoa, et cetera, et cetera. So these are mechanisms of resistance. And I then went to the several of the most commonly used and effective therapies for sickle cell disease, so for sickle cell crisis and for prevention of crisis, and found that, in fact, in each case, one of the sort of the most important mechanisms through which these drugs were effective could be tied back to the mechanisms underlying the resistance to malaria. In other words, the adaptive benefit, right, that the mechanisms underlying the adaptive benefit were essentially the targets of these drugs. Now, these drugs weren't developed with that in mind, but I thought this was pretty interesting, and I wondered whether this was a generalizable opportunity. Because after all, you might say, well, Barbara, sure. I mean, this is sickle cell, which is a monogenic disorder. It is, it has classic Mendelian inheritance. What about the complex polygenic diseases that really are the biggest killers of our species? Diseases like cancer and heart disease. And since I'm going to be talking about heart disease in a moment, I'd like to answer that question by looking at cancer. So I think many people on this Zoom are aware that there's a very famous paper. In fact, the most cited Cell paper of all time was Hanahan and Weinberg's The Hallmarks of Cancer. And in this paper, they looked at cancer, which is this incredibly heterogeneous, disease. I mean, you've got hematologic malignancies like leukemia, you've got solid tumors. I mean, it's so heterogeneous on the one hand. But they found that there were unifying characteristics that they articulated in this 2000 paper. Originally, there were 6, then there were 9 with later versions. But essentially, all of them had to do with conceptualizing cancer as a community of cells, and the disease was dysregulation of those intercellular interactions. Well, this was published originally in 2000. Soon after that, a number of evolutionary biologists and others began noticing that if you looked at the characteristics of organisms who were transitioning from unicellular to multicellular life, those characteristics were highly aligned with what Hanahan and Weinberg had identified as the hallmarks of cancer. And from this came this hypothesis that many of us I certainly for many years had sort of had this in the back of my mind that vulnerability to cancer emerged as an evolutionary byproduct, if I can say that, a consequence of selection for the adaptive advantages of multicellularity, all of the benefits that would have conferred. And in fact, if we think of cancer in those terms, right? So this is really side by side. I began wondering, well, if what happened when I compared the mechanisms underlying resistance to malaria to the mechanisms of the drugs that are used to treat sickle cell disease, I wonder if I could do make that same comparison. And so I went ahead and did that. And it was quite, this was quite a jackpot moment. And, kind of taking a giant step backward, it really makes sense that this would be the case. But what this suggested, let me explain what I'm showing here. These are 5 of the categories of cancer therapies that are effective and widely used. And I've linked each one of them to one of the hallmarks of cancer and the Hanahan and Weinberg. So what this suggests, the sickle cell and this example, is that if we have knowledge of the adaptive physiology, let me take a step back. If we can conceptualize disease vulnerability as emerging through selection for biological benefit, for some adaptive physiology. And if we can identify the selective pressures that are shaping the emergence of that adaptive physiology, and then finally, if we can identify the mechanisms underlying that adaptive physiology, right, that has been under selection, that perhaps that is a novel pathway to finding effective therapies for the disease itself. So what I'm doing is I'm making the case for conceptualizing disease as evolutionary trade-off. I'm making the case that if we can do that, that may be a way of accelerating the innovation of novel therapies. So, okay, that's an interesting idea. But in the case of sickle cell and cancer, those hypotheses have sort of existed. What if we want to find the adaptive biology that's linked to the emergence of disease vulnerability? I mean, how would we go about doing that? And so that's really the question that I wanted to answer. And my goal was to develop strategies and methodologies for, at first, for coming up with a trade-off hypothesis that we could test. And I decided to use phylostratigraphy as one of the early methodologies. Now, this is an example. This is a picture of this is phylostratigraphy from Domisett, Lozo, and Taut's paper. And what they did here, and I'm going to explain more about phylostratigraphy in a couple of minutes, but essentially what you're looking at is the x-axis is life on Earth, divided into 19 phylostrata. So over here on the left, phylostrata 1, this would be Luca over here. So this is 3.6 billion years ago. Over here, this is going to be modern times, right? And there are all kinds of reasons that they divided it into 19. I'll explain that in a moment. But they then, what they had done with this model is they created a consensus phylogeny based on lots and lots of, of species, diversity of species who had fully sequenced genomes. And so that the genes were, the origin of each of the genes could be slotted into one of these these phylostrata. And essentially what they then did was they curated cancer gene sets and they ran them through this model to show that, and just look at the red one, which is the consolidated list, that the origin of the cancer genes, so they had something like 3,000 genes, that the origin, they had these two peaks of overrepresentation, and one of them was here in this early first phylostrata, but there was this second peak right here. And this peak, interestingly, corresponded to, down here, a phylostrata in which multicellularity is emerging, that sort of 600 million year moment. So this kind of offered a kind of support or confirmation of this idea that perhaps macroevolutionary innovation, right, the innovation of multicellularity, that selective pressure around that shaped the emergence of vulnerability to cancer, right? So they took the cancer gene sets, mapped it onto the phylostratigraphy, and saw this peak of overrepresentation that correlated with this evolutionary transition, with this macroevolutionary innovation. So that kind of supported the hypothesis. And I thought, well, maybe we can use that approach for other diseases. And since I'm a cardiologist and since I have an interest in women's health, I thought we would look at, I would try to develop this methodology for cardiovascular sex differences. I think everyone is aware at this point that heart disease is the leading killer of women. If you take all the forms of cancer and combine them, heart disease still is more of a killer. And in fact, at this point, many people are aware that there are differences in cardiovascular disease between men and women. I want to add that it's important to always remember the term cardiovascular disease is often used as shorthand for atherosclerosis, which is the cause of heart attacks and strokes. But there are many other forms of heart disease that are not atherosclerotic, that have nothing to do or that are not directly due to the an arterial problem. There are issues related to the cardiac muscle, to the ventricle, to the conduction system, right? So when we're looking at cardiovascular sex differences, we see that, yes, coronary artery disease, that's really atherosclerosis. Absolutely, there are sex differences, but there are also sex differences in other forms of heart disease. And here you can see in green are some forms of heart disease that are much more common in women, for example, than in men. You see this spontaneous coronary artery dissection, 9-to-1. Takotsubo, we'll be talking about 9-to-1, HFpEF. And then there are other forms of heart disease that are less common in women, are more common in men. So what we're talking about here is our sex differences. It's not that women are necessarily more vulnerable always. That is not the case. There is really a sexual dimorphism. And that's important as a kind of nomenclature moment, because as an evolutionary biologist, but also as a physician, I know that we talk about in clinics, we talk about sex differences, but evolutionary biologists talk about sexual dimorphism. And that's important because depending on what you call it, in essence, that will shape the kind of question that you will ask to understand it. So, of course, the typical mechanistic quest of the biomedical research community is different than the kind of question that an evolutionary biologist will ask when encountering sexual dimorphism, which is, what were the selective pressures that might have shaped different female and male phenotypes? And so in the case of the cardiovascular sex differences, one of them is, which immediately when I cast it this way, that came to mind would be pregnancy. Because mammalian pregnancy is a huge cardiovascular challenge. From the beginning to the end of pregnancy, and of course, let me just say, and again, I know this crowd knows this, placental pregnancy evolved multiple times in multiple lineages. They're viviparous, reptiles and fish, of course. But we're going to be focused on mammalian pregnancy, eutherian pregnancy. That innovation is the point at which the selective pressures, I hypothesize that the selective pressures shape the emergence of the kind of cardiovascular sex differences that are apparent clinically that I'm talking about. So when eutherian pregnancy evolved, there were many adaptive benefits, right? Pregnancy protects the growing young from the environment, from predators, from hostile conspecifics. There are advantages from a nutritional delivery, from a longer gestation, maturity, survival, etc. But it comes at a cost. And the cost is that pregnancy imposes significant hemodynamic challenge, very significant. This is something you learn as a medical student very early. And certainly as a cardiologist, you know that there is a nearly in some cases 50%, but certainly most women over 40% increase in plasma volume. So the amount of circulating volume increases significantly. The cardiac output increases at least 50%. And so you have this very significant hemodynamic stressor. And in response to that, the heart muscle undergoes a significant amount of cardiac remodeling. It is physiologic remodeling. It is not pathologic remodeling. It is adaptive. It is important, but it's very significant, with the heart mass increasing in some cases up to 20%. It increases in size. It increases in function metabolically. It changes. And then following delivery, there is reverse remodeling. This selective pressure on the female, but not the male cardiovascular system shaped, I believe, two separate phenotypes. And it is the selective pressures related to the evolution of pregnancy in eutherian mammals are linked to the emergence of mammalian cardiac sexual dimorphism. And in fact, what we see in the clinics as sex biased cardiovascular disease, and I use sex differences and sex biased cardiovascular diseases, is a consequence of this selection for the adaptive benefits of placental pregnancy. So how am I going to prove that? Well, let me quickly introduce the methodology that I used, and I integrated a number of methods, but essentially I wanted to have a methodology where I could ask the question, start with the disease and work backwards to identify the adaptive biology under selection that produced the vulnerability to disease. Phylostratigraphy briefly helps us identify the patterns in the origin of genes to generate and support macroevolutionary hypotheses. It starts with creating a reference genome. You select species for this genome. You want to have a diversity of lineages. You want to have species for which there are completely sequenced genomes. You retrieve those sequences. And fortunately, we have this surge in genomes. I mean, one of the reasons that I'm excited about using phylostratigraphy is just look at the rate of rise of the fully sequenced species. I mentioned that the Earth Biogenome Project has this ambitious goal of 1.5 million eukaryotic species by the end of the decade. So this is really an opportunity. Phylostratigraphy, many of the current problems with it, the errors, et cetera, have to do with inadequate and sort of biased samples of genomes. But I think that will change over time. I'm going to really do this as quickly as I can so I can get to the guts of it. But essentially, I built a model. And I then took this model and essentially I created a model with 27 phylostrata. So here, this is, again, 3.6 billion years ago, and this is modern times. I took the protein coding genes of the human genome and using the model that I'd created from the consensus phylogeny of those species, each one of the protein coding genes of the human genome is assigned to one of the phylostrata. And at that point, that becomes the frame of reference, those protein coding genes. And then you can upload curated gene sets, disease gene sets related to your disease of interest to have a comparison of the rate of appearance of novel genes in that phylostrata. So remember, the model is created by essentially identifying the age of origin of each of the protein coding genes in the human genome. And once you have that, then you can start to make these comparisons and to look for places where there's overrepresentation, phylostrata, where there's overrepresentation of that gene set. Okay. Now, I know it's coming up on 9:35 and I said I would go quickly. So let me just get to the gut.
It's fine, Barb. No, there's no need to rush. Go through, whatever you feel like you need to cover.
Right. So then I'm going to just take a moment and say, this is the model that I built. I relied heavily on Domicellosas and Touts and Zhang. There's a number of people and RNZ who've been developing methods, but I really wanted to create something that was really accessible, usable, and where I could think of I could have a disease in mind, curate a gene set, and then look at the results and come up with a hypothesis. And so I decided to build an app. It's a Shiny app, so it's an R-based app called PhyloFastStrat, and it has multiple applications. Ultimately, this is an example of what it can do. So this is the atherosclerosis example where you see this particular graph is showing the ratio of the rate of appearance of novel genes for atherosclerosis versus the human genome. So it's the ratio of rate. So it's essentially saying where in along this history of life on Earth, where do we see a disproportionate, an overrepresentation of genes related to the disease relative to the rate of appearance of genes in the overall human genome? And when you see those peaks, you ask yourself, okay, this is really interesting. Notice there are multiple peaks. In the case of atherosclerosis, let's start with this peak right here. This is really interesting because it corresponds to the origin of vertebrates. And of course, the closed vascular system, the vascular endothelium, that is really an area that would be very interesting to look at if you're looking at the origin of atherosclerosis. So that's the process through which you have the model, you upload a gene set of interest related to the disease, you see where the novel genes related to that disease are overrepresented, and you ask yourself, over that period of time, what were the evolutionary transitions? What are the macroevolutionary innovations? Can I cast vulnerability to disease as a byproduct of selection for those innovations? So I hope that that's clear. Happy to clarify that a little bit more. Here's a QR code if you want to try. It's got lots of bugs. It should be debugged within, I think, a month or so. So even if it doesn't work, you can come back and try it again. Let me take 5 minutes and talk about Takotsubo cardiomyopathy, and just to show you the example. So Takotsubo cardiomyopathy is 9 times more common in women, disproportionate in women. It is an acute form of heart failure. This is an echocardiogram. This is the left ventricle. I think you can see that the entire apex of the left ventricle is basically hypo or akinetic. And it typically occurs under conditions of extreme physical or emotional stress. But why it is 90% female, that's the question that I decided to really focus in on. I wanted to be as specific as possible. And I wanted to really get at a non-proximate explanation. And the hypothesis that I came up with was that sex bias vulnerability to Takotsubo is a consequence of selection for the adaptive benefits of pregnancy. Now, my prediction was that if that was the case, that when I applied phylostratigraphy to a curated gene set for Takotsubo, there would be overrepresentation of the Takotsubo-associated genes and sex-biased cardiac genes in the phylostrata that were linked with the emergence of mammalian pregnancy. And so I'm not going to talk about how I curated the gene sets, but that's something that I'm also developing an app for that I'm hoping can be developed and widely used. It's my undergraduates are sort of my guinea pigs for all of these projects. That's rather fun as a teaching tool. But let me show you what I ended up finding in the case of Takotsubo. So I started with since I wanted to link, the hypothesis is that pregnancy, that the cardiac effects of pregnancy, right, have something to do with vulnerability to Takotsubo. To take a giant step backwards, I'm saying that the evolution of mammalian pregnancy imposed selective pressures on the female myocardium and not the male myocardium. And as a consequence of that, you have 2 very different phenotypes. And that over time, that sets the stage for differing vulnerabilities to cardiovascular disease. So if I was going to make the comparison, then I needed to compare cardiac genes differentially expressed in pregnancy with the Takotsubo genes. So I found there are not very many studies that look at cardiac gene expression, maternal cardiac gene expression in pregnancy. There's one in mice that I ended up using. And here you can see what this is. Again, on the red is the human genome. The blue is cardiac genes differentially expressed in pregnancy. These were the peaks of overrepresentation, and and I chose to really focus on this particular peak, right, where you see the emergence of mammalian pregnancy would correspond pretty well with this phylostrata. I also looked at these, there have been at least three now, major papers in Science on sex-biased gene expression, and I ended up making a comparison there as well. Won't go into that, but notably, we see these very similar parallel peaks over mammalia, which was gratifying. And finally, the Takotsubo gene set, again, also saw interesting peaks. And ultimately, those were the places that I decided I was going to really be focusing. So the findings were that there was overrepresentation of Takotsubo-associated and sex-biased cardiac genes in the phylostrata that were linked to the emergence of mammalian pregnancy. And this does support the hypothesis that the vulnerability, that the sexual dimorphism that we see clinically, the sex difference that we see clinically, can be traced back to the emergence of placental pregnancy. There are other features on the app that include, it's linked to gene ontology. I have it set to biological processes. I have it linked to a number of other functions. But I want to move on and talk about how to quickly test, how can you test the hypothesis? And to briefly show you, it's sort of the phylostratigraphy gives us a visual representation of the origin of disease vulnerability and allows us to put it in the context of evolutionary innovation. But it doesn't really prove anything. It provides support for, but not validation for. In order to really move past that, I wanted to develop a functional set of tools to look at co-expression. And the prediction was co-expression of cardiac genes differentially expressed in pregnancy, right? So during that cardiac remodeling, that those genes, there would be significant co-expression with the Takotsubo-associated genes. And indeed, that is what I found. I will just get to the most interesting part of it. So these are differentially expressed cardiac genes in pregnancy. In other words, during pregnancy with that remodeling, those pathways, this is what's activated, highlighting the Takotsubo genes. But this particular weighted gene co-expression analysis, WGCNA, is what I really relied on, because this method allows us to identify these hierarchical clusters of genes that are co-expressed and co-regulated, and therefore more likely to have been co-evolved. And so I used WGCNA and found that among the cardiac genes that were differentially expressed during pregnancy, the turquoise module, which was a turquoise module, this was the module in which by far there were more Takotsubo genes than any other. And this led to this led to a number of other studies. I'm going to kind of move past all of this, the heat map. But to this, which is to say that what I ultimately found was that inside that turquoise module, so that's differential cardiac gene expression in pregnancy, the most co-expressed and co-regulated genes were highly involved with biological processes related to cardiac remodeling, which is the central physiologic adaptation, cardiac adaptation to pregnancy. So that was quite pleasing, found a higher than predicted level of co-expression between Takotsubo genes and cardiac genes differentially expressed in pregnancy. And the top ranked geobiological processes that I derived from the WGCNA were most enriched for TTCM genes, central to cardiac remodeling and reverse remodeling, precisely what occurs during pregnancy. So I will end there. I just want to show you one interesting association. There are all kinds of implications, but I ended up getting really interested in this linkage. Now, I have to say that the connection between risk of cardiovascular disease later in life and selective pressures related to pregnancy, that's a new hypothesis. I'm excited to say that. But I also became interested in parity. In other words, if in fact the cardiac pathways that evolved in the maternal myocardium to adapt to the hemodynamic challenges of pregnancy, if those are the same pathways that are significantly involved in Takotsubo risk later in life, then does parity influence the risk of Takotsubo? And we have not yet published this. I'm working with Inference, which is a data company that had access to the Mayo Clinic, this large data set. And we found that parity in their population, this is an AI company, so there's massive amount of patient information. We found that parity actually is a risk factor for Takotsubo, and that in fact there's a dose response curve between the number of offspring and risk of Takotsubo, which is pretty interesting. So I am going to just go zoom, 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 all of these things, and end with the fact that there is a disease that's rare, but that can actually lead to mortality in younger individuals. It's called long QT syndrome that can lead to a cardiac arrhythmia. And parity, great grand parity, which is over 9 deliveries, right? You see that it places patients at significantly increased risk. This won't mean anything to you if you're not a cardiologist, but this is a very long QT that sets someone up for an arrhythmia. And I started looking at that and I thought, well, I wonder if it's possible that the cardiac effects of pregnancy actually are a constraint on human fertility. And so I had to go look at, this is kind of at just the mean number of live births in the Chimani, and of course it was 9.1, which I don't think that means anything, but I thought it was kind of interesting. Anyway, I'll wrap it up, and I would love to hear questions.
Great, thank you, Barb. It's very exciting. Thank you. It was great to hear about this work. Thanks so much for sharing. We have a number of questions that are in the chat. I want to invite people who've posted questions there to raise their hands, and others as well to raise their hands. I see that Jillian has her hand raised, so I'll call on her first. And let's see, Jillian, are you able to ask your question, or do I have to give you permission?
Yeah, can you hear me? I can't seem to turn on my camera. But yeah, so that was a great talk. Thanks, Bob. And I'm going to act a little bit like devil's advocate here. So is it possible that the heart in humans is just plastic? And so what you see here is more precisely because you said that there's an effect of parity as well on the risk for TTCM. But if males experience something similar, in other words, would they be able to react in a cardiac manner in the same way? And I'm thinking of an example here. It just struck me when I was reading some time ago about what happened to the English men that climbed Mount Everest. When George Mallory's expeditions went up there, they took a physician with them, and their hearts became enlarged during climbing. And I'm just wondering if you could look at that as a sort of similar response that you might get during pregnancy, because of course these men who were not physiologically adapted to high altitude had to deal with being at high altitude, and so had these experiences. And
yes,
yeah, I guess
so let me address that. Yeah, it's a great question.
Just one other thing, sorry, just one other thing too I wanted to raise. Is it also possible, again playing devil's advocate, that the kinds of stresses that might induce TTCM in women, which I think is also referred to as broken heart syndrome, might be culturally specific as well, and that women might have a more extreme reaction because, for example, if they're broken up with or experience, a separation, they're not in a position, perhaps financially, to cope, and so they experience greater stress than a male in a similar position.
Yes. All right. So great. So let me quickly address the first and then the second. There is, let's move away from humans to other eutherian mammals. There is very clear data that the male ventricle is less adapted to volume overload than the female, and the female ventricle is less adapted to pressure than the male. In other words, there's a trade-off between pressure adaptedness and volume adaptedness. This is something Rob Shave is on the call. He and Dan Lieberman wrote a paper about actually chimps that looked at pressure and volume adaptedness across different species interspecifically, but even intraspecifically, there is definite sex-specific, in non-human, eutherian mammal sex-specific volume adaptedness in females that exceeds males. So much so that in experimental conditions, when they induce, they rip up the valve, the aortic valve, so the mice have severe aortic regurgitation. The females tolerate it for weeks longer than the males. The males develop heart failure, elevated and pulmonary vascular congestion. So there's no question. There's no question that the female ventricle is more volume adapted than the male. The second is that in instances where different investigators have manipulated the sex hormone. They've overectomized the females. They do lose some of the volume adaptedness, but not all. And males given estrogen do not become more volume adapted. So there's undoubtedly lifetime effects. Parity has an influence, but these are intrinsic and not small differences in volume and pressure adaptedness. And the final point I want to make is that it's hard to overemphasize the importance of the significance of the hemodynamic challenge of pregnancy. It is when we're talking about a 50% increase in cardiac output, right? Cardiac output is stroke volume times heart rate. That is a massive increase in workload. It is the sort of central athletic event in a female mammal's life. And I think we've not been paying enough attention to that. We're talking about cardiac remodeling where we're with 20% increase in cardiac mass. So even though undoubtedly there are going to be cultural influences on that, without a doubt, it's just a biological reality. The one other issue I want to make about Takotsubo is that for a long time, Takotsubo was first identified in Japan, actually. It's called Takotsubo cardiomyopathy because the shape of the ventricle that I showed you apparently looks like the octopus fishing pots, and the Japanese cardiologists recognized it in the cath lab when they did a ventriculogram. And originally it was felt to be only emotional stress. Now we understand that physiologic stress, sepsis, there were a number of cases with COVID. So whether it's emotional or physiologic stress, it's a high catecholamine environment that appears to trigger this, but some individuals are more vulnerable than others. So I don't think that it is at least cross-cultural in that it occurs in, it was first identified in Japan, but it's been seen all over the world. And while there may be difference in how women and men experience stress, I'm sure that there are. I think we have to do better than just attribute that 9-fold increase in female vulnerability to the cultural stressors related to being a woman.
Okay, great. Thanks for that answer. And I'm going to go to Catherine because I had the same question Catherine had, and I'll just add to it after she asks her question. Go ahead, Catherine.
Oh, great. Thank you, Charlie. Can you hear me?
Yes.
Okay, terrific. Thank you, Barbara, for your talk. Your work is so interesting. My question is, given your approach to interpreting the overrepresentation of genes in that phylostratigraphic record as being evolutionarily meaningful, how are you thinking about the underrepresentation signals that we see there?
Yes, I love that. Yes, well, thank you for that question.
Sure.
And let me just take a moment and say that the reason the overrepresentation, even that we think it has meaning, is based on how we understand novel phenotypes emerging, right? That novel genes appear, they're integrated into pre-existing biological pathways, producing new phenotypes that, under selection. And then in some cases, we have a novel phenotype that may become an innovation and, something major. So that's the reason we're looking for overrepresentation in the first place. Now, the phenotypes can emerge from, you could say overrepresentation or underrepresentation, and certainly gene loss is associated with novel phenotypes as well. So I would say both would be useful. The model that I am showing you is very crude. I mean, first of all, it's, we're talking about 27 phylostrata. It's so broad, hundreds of millions of years shown. But it's an idea. And there are many, many problems with phylostratigraphy today. Many. The other point I want to make is that you saw there are many peaks of overrepresentation. I mean, why choose one versus another? And I would say each one of those peaks of overrepresentation represents at least one hypothesis, that phenotypes are composed of multiple, elements of biological content. So it's just it's a way of beginning to develop a trade-off hypothesis.
Great, thank you. Appreciate that. It's good to
And
hear how you think about things. Charlie?
My additional question was just how do you decide what's an overrepresentation on those plots? And I think you just answered it. It's, you're just looking graphically for these, and wherever you see a change, you're saying that's a point where you can propose a new hypothesis.
Yeah, and there are standards. I used integrated methods, Damazot, Lozos, Arendzi, these different folks who've come up with a standard, a mathematical standard, and there's a line of significance. But ultimately, it's a new technology. It's a new approach. It's an idea.
Okay. Dawn, would you like to ask your question?
Sure. Well, first of all, thank you for welcoming outsiders. I'm a pulmonary critical care physician. My research focuses on sex differences in the lung, especially network approaches to sex differences in the lung. And one of the challenges we have using existing methods is it doesn't include allosomes. So as you are looking at these evolutionary models in the heart, are you including X and Y when appropriate in this evolutionary context? And yeah.
So
yeah.
So I'm really I'm not looking reductively even in that sense. Now, one thing, since you're a pulmonologist and critical care doc, is that the idea that the hemodynamic challenge of pregnancy would constitute a selective pressure that shapes the emergence of cardiovascular sex differences, I think that applies to probably sex differences in any organ. So whether it's, just in terms of vascular, the smooth muscle and the vascular. So whether it's pulmonary, whether it's kidney, whether it's all of these sex differences, I think volume has been the adaptations to pregnancy, particularly volume. I think it's an opportunity to understand some of the phenotypic differences and vulnerability. We have a project. I have a group of nephrologists who are looking at sex differences in kidneys based on kind of a volume hypothesis, a trade-off hypothesis.
Okay, great. We're almost out of time. I want to try to fit in two more questions. Jay, go ahead.
Hi, Barb. Great talk. Can you tell us a little bit, if you look across different species, what the effects are on the number of offspring that a female may have in any one litter, mice versus elephants, for example? And also the amount of time that a pregnancy lasts, mice 20 days versus an elephant a year. To what extent do we see the development of Takotsubo across those different kinds of traits?
Right. So first of all, we don't even know whether Takotsubo occurs spontaneously in other animals. There are a couple of animal models. I suspect there's a connection to capture myopathy. I can't prove it. So I can't give you any information about, litter size and speed, but I can say that it's an interesting opportunity to further prove or disprove the hypothesis. If in fact the emergence of eutherian pregnancy and the plasma volume expansion and hemodynamic challenges of pregnancy shape these differing selective pressures on males and females, okay, then yes, there are these that could be the reason we have these phenotypic differences. If that's the case, as we go leftward on mammalian lineage through marsupials, through monotremes, as we move into other taxa, mammalian taxa, where there's less, and then in the case of monotremes, no pregnancy-associated volume expansion. So marsupials have for the most part, more pouch time than, than uterus time. So that we should see cardiovascular sex differences decreasing. In other words, there should be a kind of dose response curve, in a sense, between the amount of cardiovascular sex difference and the amount of volume expansion in pregnancy. And ideally, monotremes, we would see none. Now, having said that, that's not true either, because of course, there are cardiovascular sex differences in reptiles and in fish. One of the most interesting, there's a teleos that the heart rates of male and female embryonic fish are significantly different, that certain fisheries use the heart rate difference to sex the fish before birth. So certainly, even if eutherian pregnancy was a significant selective pressure that shaped the emergence of these different cardiac sex differences, that was acting on pre-existing. There were constraints related to sex differences in the vertebrate heart that even predated the emergence of eutherian pregnancy.
Thank you.
Okay, thanks. And the last question goes to Ronnie. Go ahead, Ronnie.
Hi, Barbara. Great talk. Thanks for that. I'm wondering if you know whether eutherian species in high stress environments like extreme climates show greater variability or any adaptations in, say, pregnancy-related genes that could influence cardiovascular resilience?
That is a really interesting question. I don't have an answer to it. We'll say that the connection between climate and vascular function, placental vascular function, we know now, for example, that that heat and other climate-related changes are inducing prematurity in a number of communities around the world, premature birth in communities around the world, and there's a significant shared gene groups between placental and cardiovascular remodeling. So there's that link, and these differences, these cardiovascular sex differences in vascular compliance, autonomic tone, and they are very significant. Those exist even before puberty. So those are intrinsic, those are deep, and those I think are, I can't say that they're more fundamental, but the fact that they exist earlier in life, I think that they are probably shape phenotype in a very significant way. And they're influenced by, of course, by climate and by volume status.
Got it. Thank you.
Sure.
Okay, thank you very much, Barb. Thank you, everyone, for attending. I'm sorry, Albert and Barry, that we weren't able to get to your questions. Obviously, there's a lot of interest in this talk, and that's great. Stay tuned. We have a bunch of talks coming up in 2025, including a series of about 3 talks on evolution-proofing therapies and treatment. And that'll be a great lineup. We also have a couple of EMPH, Evolution Medicine, and Public Health articles that are going to be featured in 2025. So stay tuned. We'll have a great lineup in 2025, and it's great to see everybody. Thanks.
Thank you.
Thanks, Barbara.
Thank you.